Okay. Good afternoon. Let's start with the next session. And the first speaker is Jennifer Doudna. She did her postdoc with Tom Check, really being interested in RNA structure. And then she went to Yale, her faculty position. And in 2002, moved to Berkeley, where she still is. Now, Jennifer has been interested really in catalytic function of RNA, and she got interested in something rather, I think, esoteric and maybe specialized, namely the immune system of bacteria, until she realized a few years ago that this CRISPR-Cas system, which was mentioned today, is really a system to gene edit the genome of mammals and other animals in an unprecedented way. And this really has led to an explosion in the field with a dozen papers probably each month being published. And probably it's fair to say that this technology will revolutionize biology the same way as PCR did. So Jennifer is a recipient of a number of prizes. One is the Eli Lilly Award 2013 and the Lurie Award this year. She's an investigator of the Hughes Institute. And her talk is the biology of CRISPRs, from genomic defenses to genetic engineering. Jennifer.
Well, good afternoon. Thank you very much for that generous introduction, and I'm really delighted to be included in this illustrious symposium. I'm learning a lot and seeing a lot of good friends as well, so it's been great to be here. So what I want to do today, as Rudy mentioned, is I'd like to discuss some of the work that we've been doing over the last few years, very basic biology to try to understand how bacteria fight the flu. And I'll just tell you that we got into this because of our interest in microRNAs, and in particular in RNAi processing pathways in human cells. And my lab has been working in that area for at least a decade since we moved to Berkeley. And a few years ago, it came to my attention that there was a predicted pathway in bacteria. At the time, there was no experimental data for this, suggesting that bacteria might use small RNAs to recognize foreign invaders into their cells, but perhaps, and I thought, perhaps they're using these RNAs in a fashion similar to RNA interference, but with entirely different protein machinery. And I thought this would be an incredibly interesting thing from just a very basic biology perspective to study and to compare to RNAi. And so that's really how we got into these CRISPR systems that I'll tell you about. So this is a cartoon that just illustrates in a very simple way the way that these pathways actually work. And so what had been noticed in around 2005 by several different groups was that in a lot of bacteria, about 50% of sequenced bacterial genomes have CRISPR loci. And what was interesting was that these consist of 40 base pair repeated sequences shown in these black diamonds. And these three groups pointed out that in many of these loci, you could actually find exact matches between the intervening or spacer sequences in between the repeats that matched sequences found in bacteriophage, which are the viruses that infect bacteria, or sometimes from plasmids. And so that sort of suggested that these might be signatures of some kind of acquired immune system in these organisms that might function together with a set of genes called Cas, or CRISPR-associated genes, that together might operate to allow these bugs to acquire sequences from their invaders and then use them to defend against infection. So in fact, this is exactly the case. And so there were a series of papers in the mid to late 2000s from labs doing mostly microbial genetics that suggested that, in fact, the way these systems function is they allow organisms that have a CRISPR locus to detect foreign DNA that gets into the cell from either a viral infection or a plasmid transformation. They can excise a short sequence of DNA from that material, and they incorporate it at one end of the CRISPR locus. And then that information provides the template to synthesize a precursor RNA molecule, which is subsequently processed into smaller RNAs that assemble with proteins encoded by the Cas genes to make interference complexes in which the information in the CRISPR RNA is then actually used to recognize foreign DNA and lead to its degradation. So we started investigating the biochemistry of this and try to understand the molecular basis for this pathway. And I just want to tell you very briefly that in many of these systems, and there are multiple types of different CRISPR systems, it's clear that the interference pathway operates through a series of genes, gene products, proteins that come together and assemble with CRISPR RNA into a fairly large targeting complex. And so I'm just showing you here an example of the type, what's called the type 1 CRISPR system, an example found in E. coli. And we know that in this type of CRISPR pathway, there are 8 genes involved in the CRISPR system. 2 of them are responsible for integrating new sequences into the CRISPR locus, and the other 6 over here are part of the actual interference step. 5 of these genes encode proteins that assemble into a large complex called cascade that, together with the CRISPR RNA, is able to recognize foreign DNA and then recruit a separate endonuclease called Cas3 that leads to degradation of the foreign DNA. And so we've done a lot of work over the years on this pathway, and I'm not going to say much more about it today, because I really just wanted to contrast that with what happens in the type 2 CRISPR systems, exemplified by the system found in a human pathogen called Strep pyogenes. And in this organism, in this type of CRISPR system, there are 4 genes involved in the Cas, the CRISPR-Cas system, 4 Cas genes. And what was shown genetically was that 3 of these are actually involved in integration, again, the step that acquires new sequences into the CRISPR locus. And that left just 1 gene, a gene called Cas9, that was found to be genetically important for DNA targeting and was speculated to be potentially involved in the degradation of foreign DNA. And so we set out in 2011 to collaborate with Emmanuel Charpentier's lab to discover the function of Cas9. And so in a series of experiments that were done in both of our laboratories across thousands of miles, thank goodness for Skype, and her student Krzysztof Chylinski and my postdoc Martin Jinek worked together to investigate the function of this protein. They started with purifying this protein and then investigating its activities with different kinds of nucleic acids. And what those experiments showed was that Cas9 is actually a very interesting protein that uses not 1, but actually 2 separate RNA molecules to guide the recognition of foreign DNA and lead to a double-stranded break in the DNA. So the blue cartoon here shows the outline of this protein, Cas9. It has 2 active sites, 2 catalytic centers, one homologous to RUB-C and the other to an H&H nuclease domain. And what Martin Jinek's experiments showed was that each of those catalytic domains is responsible for cleaving one of the DNA strands of the double helix in the region that is complementary in sequence to the guide portion of the CRISPR RNA, which is this RNA molecule right here. So there's a 20 base pair interaction between the guide sequence and one strand of this DNA double helix that leads to a local unwinding of the DNA. Very importantly, it has to be next to a short motif called a PAM, which for this enzyme is a GG dinucleotide motif. And then also very importantly, this RNA doesn't function alone. It actually requires a second RNA that Emmanuel's lab had previously discovered called the tracer or trans-activating CRISPR RNA. This RNA is actually important for processing of CRISPR RNAs in organisms that have this type of CRISPR system, but it doesn't go away after processing. It remains associated with the CRISPR RNA to form an interaction, a structure here that recruits the Cas9 enzyme. And without that second RNA, the system doesn't function. And so once we had that information in hand, it was actually possible to look at this. And I remember a day when Martin Jinek was in my office and we were looking at his data and we had this sort of sketched up on my whiteboard. And we realized that we could probably turn this into a single transcript, a single RNA molecule by simply connecting the ends of the RNA to make a chimeric molecule, RNA molecule, that would contain the targeting information and the structural information necessary to bind to Cas9 in the same molecule. And that was sort of the moment when we looked at each other and said, wow, if this works, this will be really exciting potentially for genome engineering, because we thought here we would have a system that was a two-component system, a single protein that could be directed to essentially any site in a genome by simply changing the sequence of a guide RNA molecule. And this was just an initial biochemical experiment testing this and showing that, in fact, we could direct Cas9 to sites that we targeted by designing different guide RNAs, in this case 5 different guides shown by these red lines that targeted sequences chosen within a plasmid that we got from Barbara Meyer's lab. And we showed by double digesting this plasmid with the guide RNA directed Cas9 together with a second restriction enzyme cutting just upstream of that region that we released fragments from the DNA corresponding to the sizes we predicted based on where these guide RNAs should be enabling Cas9 to cut. So that was really an exciting moment, and then that this has led to some really exciting developments in various areas of biology now. So we know that this system is useful for making gene disruptions, and this works by simply introducing a double-stranded break into DNA, and by very well, or at least fairly well known pathways in many cells that have repair machinery endogenous to the cell, they're able to repair these kinds of breaks by either generating a fusion of the DNA through a process called non-homologous end joining, or by introducing new sequences at the site of the break through a process called homology-directed repair, if there's a donor DNA molecule around. And so this has been used now to introduce changes to the genome in a number of different systems, organisms, and cell types. And importantly, this is a technology that's not only interesting for making covalent changes to the genome, but it's also been possible to use a catalytically inactive form of the Cas9 enzyme for transcriptional control. And there, I think what's exciting is that it's useful not only for transcriptional repression, but also for transcriptional activation. And this is a lot of work that's been done at UCSF by Stanley Chi and Jonathan Weissman and Wendell Lim, for example. And then even more recently, using the same kind of strategy, not for regulation of the genome, but actually for imaging of the genome in live cells. Work by Bo Huang at UCSF with collaborators has shown that you can use a fluorescently tagged version of Cas9 for that sort of imaging. So it's clearly a technology that's very useful because it allows targeted localization to the genome, where you can either then introduce covalent changes or you can recruit factors or proteins to that particular site. So in my lab, we've been just keenly interested to continue understanding how this works. We'd like to understand the basic mechanism of this very interesting protein. And then, of course, we'd like to use that knowledge to continue to explore different applications of this technology and other kinds of things that we can do with it. So what I very briefly want to do is to tell you a little bit about what we know about how Cas9 finds target sites within genomic DNA. I'd like to tell you a little bit about how we think that the Cas9 protein is able to open the DNA double helix. And then at the end, I'm going to show you some very new data that I think is very exciting, that we're able to use our mechanistic insights to now use Cas9 in a completely different way. So first of all, how does it find sites within the genome? And so this work has really largely started with a collaboration between my lab and Eric Green's lab at Columbia, in which we used a system that Eric has set up called DNA curtains, in which he was tethering single molecules of phage lambda DNA. These are 48 kb DNA molecules that are visualized by an intercalating dye. They're tethered at one end. And then we have a quantum dot labeled version of Cas9 that is programmed to recognize different sites in the lambda DNA genome. And you can see in this little video here that we can toggle between having the buffer turned on. The buffer is flowing across the slide from top to bottom, so the DNA is extended. When we turn off the buffer, the molecules snap back up to the origin. And that's useful for determining which quantum dot labeled proteins are actually on the DNA and which ones might be just stuck to the slide. And so you can see in this sort of experiment that we get a lot of molecules that are lining up where we expect them to, roughly, in the lambda DNA based on the guide RNA that was used in this experiment. And if we do this with multiple different guides, we can actually get a plot that looks like this. So we get very nice targeting in this sort of system. So Sam Sternberg, a student in my lab, together with Cy Redding, a student in Eric Green's lab, worked together to use this system to measure a number of the properties of Cas9 in its search for target sites in DNA, and what happens when it finds, also what happens when it interacts with non-cognate sites. And so I just want to show you one experiment that, so I'm going to summarize the results from those single molecule data, but I also want to tell you that this was very useful for us in terms of coming up with hypotheses that could then be tested in bulk assays as well. And so a lot of the biochemical work that Sam did while this was all going on together with Eric's lab was to test ideas in bulk assays. And so one of those ideas that came out of this was we recognized early on that the PAM sequence that's adjacent to target sites that are recognized by Cas9 is actually very, very critical for Cas9 to get on to the DNA. And we could see that in the single molecule experiments. And we wondered if it was important only for recruiting Cas9 or if it might also be important for something else. And so this was an experiment in which Sam tested a double-stranded DNA target sequence. So the PAM is shown in this yellow box, and the target site is here in red. And you can see we get very nice cleavage of this DNA substrate by Cas9. But if we use just the single strand of DNA, just corresponding to the sequence that base pairs with the guide RNA, we get normal binding to Cas9, but we get virtually no cutting in that kind of reaction. So that was kind of curious. We thought, well, it's binding, but why is it not getting cleaved by the enzyme? And so Sam started adding back short DNA oligos, thinking we thought initially we might need to have a completely double-stranded substrate for the system to work. But we actually got a very interesting result when we added back just short DNA molecules that recreated the double-stranded nature of the PAM, so included the PAM nucleotides GG right here. As soon as we added back an oligo that included those Gs of the PAM, we got very nice cleavage now of that single-stranded DNA. And this is just a plot showing that we get all four of these substrates bind with virtually the same affinity to Cas9. So it's not a binding defect, it's really a catalytic defect. And so we think that really PAM binding, in addition to recruiting Cas9 initially to the DNA, is also actually important for activating catalytic activity. And so this is kind of a summary of what we think the mechanism of DNA interrogation really involves, which is binding occurring first at PAM motifs. I didn't mention this before, but we also know that, after DNA cleavage, those DNA products remain very tightly associated with Cas9. And we see no evidence for any substrate turnover, which might have interesting implications for the use of this system in cells for cellular editing. And then also, as I just showed you, we think that PAM binding triggers catalytic activity of Cas9, potentially through a conformational change in the protein, something that I'll come back to in a few minutes. So how does Cas9 open the DNA duplex? So there are a few lines of evidence that suggest that it really does actually open the duplex. It's not a triple helical formation. It's actually an RNA-DNA hybrid that occurs, and then and the other strand of the DNA is displaced locally, allowing sort of local unwinding of the DNA. And yet this is an enzyme that doesn't use ATP. So, how does that work energetically? And I want to show you just one biochemical experiment to sort of get at this. And we know that there's really an interesting coupling between the interaction of the protein with the PAM and then the initial steps of interaction between the guide RNA sequence and the target site in the DNA. And this is an experiment that kind of gets at this. So if you look at the top panel first, these are native gels where we're using radiolabeled DNA, and we're asking how well do those DNA targets bind to a Cas9 guide RNA complex to form a sort of a ternary complex that we can see in a gel system like this. And so if we do that experiment with a perfect DNA substrate, we get a very nice shift forming this ternary complex. But if we do the same experiment, and now we have a two base pair mismatch in the two base pairs immediately adjacent to the PAM in the target site of the DNA, we get very, very poor binding. But what's I think very interesting is that we can actually restore that if we keep the same sequence of the bottom strand, so we still have a mismatch with the guide RNA on this bottom strand, but now we also make a mismatch in the DNA itself, right? So there's a little artificial bubble in the DNA. We now see really nice binding. And you see the same thing reflected if you look at cleavage assays down here. This one cuts very well, this one very poorly, and then this one, cleavage is restored again. Right? And so it really suggests that there's an interesting, I think, energetic coupling between initial binding and the ability to unwind the DNA adjacent to the PAM and allow base pairing with the guide RNA. And that's something that we're very actively investigating right now, is to really understand the energetics of that process a bit better. Now, one of the clues to this has come from structural studies done in our lab and other labs. And so in collaboration with Eva Nogales, we did some fairly low-resolution EM studies of what happens to the Cas9 enzyme as it assembles with guide RNA and then with a DNA double helix. And what we found, and I'm just showing you the cartoon version of this, but it is published if you want to have a look at it. What we know from those studies is that Cas9 starts off in sort of a closed state. It's got two very clear structural lobes to it. One lobe, which is shown in blue in this cartoon, is the catalytic portion of the protein, so it has the two active sites over there. And then in gray is what we call the alpha helical lobe. This is the part of the protein that's actually, we think, very important for guide RNA binding and eventually for DNA binding as well. When we add the guide RNA, we can already see by EM that there's a rearrangement in the protein that clearly separates the two lobes of this enzyme and creates a channel down the center where the DNA actually binds once this assembles with a substrate. So, kind of remarkable to me that, even at fairly modest resolution by EM, we could see these very interesting rearrangements in this protein that hinted at something very, very interesting going on as it assembles with nucleic acid. And I want to just show you a movie that illustrates this now on a more sort of molecular level. So this is now comparing two crystal structures of Cas9, starting with the structure you're looking at here, which is in the unliganded state, so at the starting state, and then morphing to a structure published recently by Nareki's lab in collaboration with Feng Zhang, in which they have a guide RNA and a single strand of DNA associated with the enzyme. And the change in the structure is really very interesting, and it parallels really almost exactly what we had seen using electron microscopy. So this starts off with in the unliganded form, and in the colors here are the active sites of the protein. Notice this very long green helix, which is critical for nucleic acid binding, and we knew that already from various mutational studies. And as it associates with nucleic acid, you're going to see a very large rotation in this domain here that swings this around and creates the cleft that actually binds to the RNA-DNA hybrid on this side and to the rest of the RNA handle over here, and then extending down through the protein to the 3' end of the RNA here. So just an amazing rearrangement of this enzyme as it actually assembles with nucleic acid. It's going to go back one more time, and if you notice, if you sort of look at the base of this long green helix, you'll see that the hinge is really right here, right? So it sort of allows this rotational change between the two lobes of the protein. And so, of course, we can't help but wonder whether this isn't partly what is to explain, what explains this ability of the enzyme to open the DNA double helix without requiring ATP hydrolysis, for example. So this is our current model for the way that we think about the way that Cas9 interacts with DNA, genomic DNA, for example, and that is that we see no evidence by the single molecule studies that it's a processive enzyme. Instead, it looks like it's a dissociative mechanism. It's on and off the DNA very, very rapidly, except for regions that are rich in PAM sequences. That slows down the off rate of the enzyme, and we get slowest off rates when we have PAM sequences adjacent to a cognate or almost fully cognate target site. And when that occurs, then it actually, we think, gives time for this local unwinding of the helix adjacent to the PAM, and then there's a directional melting in of the RNA strand as the DNA opens up to give eventually a fully competent complex where the DNA actually gets cut. So the final thing, in just the last couple of minutes, I would like to share with you some ideas that we have about how we can take that kind of mechanistic understanding of how this enzyme works and use it to redirect the activities of Cas9. So one of the things that we asked ourselves early on was, why does this target DNA? Why doesn't it target RNA? Why doesn't it work like RNAi? And yet when we, and this has been done in other labs too, tried to get Cas9 to cut RNA, it never worked. It would bind, not very well, but it never really cut the RNA well at all. So the kinetics were very, very poor compared to what we saw for double helical DNA. But once we had the information that I showed you, the experiment that Sam had done with a single strand of DNA, and activating that for cutting by adding back a short DNA oligo that has the PAM nucleotides on it, we thought, well, maybe we can actually activate Cas9 to cut RNA substrates if we provide a PAM oligo to mimic the PAM that it would normally be finding in a double-stranded DNA duplex. And I'm going to show you that that actually works very nicely, okay? So what you can do is, and so this is a cartoon at the top here that diagrams the experiment. So what we have is we're basically asking, can we cut a single-stranded RNA target sequence that is able to base pair with the guide sequence in this Cas9 RNA complex under different conditions. And in particular, in this experiment, we're doing this by adding back what we call a PAMmer. It's a DNA oligo that includes the PAM nucleotides at one end to form a structure that we kind of imagine might look something like this. And so if you do this experiment, what you find is that, so this is, if you look down here, this is a denaturing gel system that shows cleavage of these different substrates that are pictured here. This is just a normal double-stranded DNA target sequence, so you get very nice cutting with Cas9. If we try to cut a single-stranded RNA, it's a very poor substrate. It essentially doesn't get cut well at all. But if we add back two different versions of this PAMmer nucleotide, we can get really nice cutting of the RNA. And note that if we do this with a double-stranded RNA substrate that includes the PAM nucleotides but in the RNA form, that's absolutely not cut by the enzyme. We don't think it has the ability to unwind that RNA duplex. And then this is just showing that this is actually really fully programmable, just as we see for double-stranded DNA targeting. So this is an experiment in which we used 3 different guide RNAs to recognize different sequences in RNA. And what you can see here, maybe, this is again a denaturing gel system, is that we get very nice cutting of just the RNA that is directed by each of these guide RNAs that are used with the Cas9 enzyme in each case, and no cutting, no detectable cutting at all of these other ones. And this is just showing a quantification of that. So we've been doing a lot of experiments with this and testing how we might be able to use it in cells. We're interested in using it for imaging. I'm very excited actually about being able to use it for tagless RNA detection in either live cells or in cell extracts. And we have some very recent data. I didn't even have time to make a slide showing that we get very nice tagless detection of RNAs also in this programmable fashion. So stay tuned. I think this is a really interesting enzyme that just kind of continues to evolve as we understand better how it works, and we can figure out ways to use it that extend and expand on what it does naturally in nature. So to conclude, basically, we know that this is a programmable double-stranded DNA endonuclease. We know that the binding of the guide RNA basically activates this enzyme for DNA surveillance, probably through this structural rearrangement that occurs in the protein, and we're working hard to understand that better. We think that the PAM is important both for recruiting this complex, but also for initiating a directional melting in of the DNA, and ultimately to catalytic activation. So there's sort of multiple levels on which this protein is regulated, and cells are able to control its activity until it's really engaged with a bona fide target. So I'm going to conclude just by acknowledging a wonderful group of people in my lab. This was taken recently up at Tilden Park in Berkeley. And great people that have been involved in the project. So Martin Janek kind of started it all with us working on Cas9 and is now running his own lab at University of Zurich, where he continues to work on Cas9 structural biology. Sam Sternberg is a graduate student who did all of the single molecule work that I mentioned today, as well as a lot of biochemical work together with Cy Redding, a student in Eric Green's lab at Columbia. And then all of these folks have been involved in different aspects of the system, in particular Mitch O'Connell, who's been doing work on the programmable RNA targeting by Cas9. And I want to also just acknowledge wonderful collaborations that we have that are, a lot of which are sort of ongoing currently. Thanks a lot for your attention.